So, hello everybody. It's a real pleasure to moderate together with Rosario today's BIH lunch lecture, and we have a special lecture today because we have the pleasure to have with us Cherilyn M, scientific editor at Cell, who will give us some insights. But actually, Rosario will introduce her in more detail in a minute. But before I hand over to Rosario, I would like to announce some housekeeping remarks. Please turn off your webcam and microphone in order to really assure that everything goes smoothly. And later on for the Q&A session, please send your question or post your questions into the chat. And just as a side note, the lecture will be recorded for the website. So with this, I hand over to Rosario to introduce Cherilyn.
Hello, thank you, Birgit. So, as Birgit said, our speaker today is Dr. Sirois. She's currently a scientific editor at the journal Cell. Cherie started her scientific career as a laboratory research scientist and obtained her PhD in biomedical sciences from the University of Massachusetts Medical School in the United States. Then she did her postdoc at the Centers for Therapeutic Innovation in Boston. Also, Dr. Sirois was a professor of immunology and microbiology at the Universidad de las Américas in Quito, Ecuador, and also at Springfield College in Massachusetts. Then in 2018, she joined the editorial staff of Cell Press, and she's currently based in Cambridge, Massachusetts. And today we have the pleasure to have her here in BIH giving us some advice on what do journals really want. Dr. Sirois, thank you for being here. Now the stage is yours.
Thank you, Rosario, and you're welcome to call me Sherry. It's really a pleasure to be with you here today, and thanks so much to Dr. Sawitzki for the invitation. And I hope to speak for about 30 minutes, maybe a little bit less, and then have lots of time for questions. So I look forward to hearing from all of you. All right. So as Rosario mentioned, I'm an editor at the journal Cell, and Cell is part of Cell Press, where we have 28 different research journals in addition to Cell, 27 plus Cell. And so we have journals that cover lots of areas of science, including translational and clinical sciences, which I know is of interest to many of you, as well as basic science and lots of disciplines and physical sciences now as well. We also have journals that focus specifically on reviews. Those are the trends journals. And Cell Press is part of the publishing family of Elsevier, which of course has a great many scientific journals. And all of the journals at Cell Press are essentially thinking about papers in regard to two things: the scope of the journal and the conceptual advance provided by the paper. And so there I'm showing you a picture of a microscope. And of course, we don't use microscopes at the journal, but there you can see the viewfinder of the microscope. And it just is the way you think about a sample. Everything that's within the field of view is within the scope of your analysis, right? And so each scientific journal has a scope and an area of view that it's interested in. And that differs for each journal. And so when you're thinking about where to send your paper and whether your paper would be a good fit for the journal or the journal is a good fit for your research area, it's important to think about the scope. So how could you know that? How can you know what a journal is interested in? For Cell Press journals, I would encourage you to take a look at the website. The website for each individual journal. Here I'm showing you an example from Cell and from the journal Neuron. Their website will have a statement of what the scope of the journal is. And so I know there's a lot of text there, but maybe you can see the first sentence says, Cell publishes findings of unusual significance in any area of experimental biology, and then lists some of our areas of interest. So Cell is a broad scope journal. We are interested in all kinds of science, particularly biological sciences. And that phrase of unusual significance is quite important to us. So Cell is quite a selective journal, and I'll talk to you more today about what it is we're looking for. But in general, what we're interested in are the studies that are particularly unusual and telling us something that really changes the way we think about a field or about science in general. In thinking about the scope of a journal, you can also take a look at what the journal has been publishing in the last few years and see, are they publishing things in my area? Even if a journal isn't, if it still hasn't published in your area for the last few years, that doesn't mean we wouldn't be interested. But oftentimes you can get a good idea of the journal's scope by looking at their current publications. And if you're still not sure, it's always fine to ask an editor at the journal if they're interested in your study. So talking to editors is a really good way to decide what the best course is when you're getting ready to submit a paper for publication. So this is a slide showing the photos of my colleagues at Cell. There are currently 16 scientific editors at Cell. These are the folks who will read your papers if you submit a paper to us, including me. And we're all trained scientists. Everyone here has a PhD. Many of us did postdoctoral work. Some of us worked as independent scientists, as PIs, and had labs before becoming editors. So we understand the work that you do, and we ourselves are scientists and think like scientists. And so just like every scientist sort of has a specialization of what they focus on in their work, we have a scientific background. I was trained primarily as an immunologist and a microbiologist. Some of my colleagues were neuroscientists, cell biologists, molecular biologists. But now we are all scientists who are essentially specialized in the editorial process. And so you see there's an arrow with a question mark. Now what our job is knowing what sorts of things we should publish and sell. So we're scientists devoting to the specialty of curating scientific work. So it's sort of a strange word to use, or curation. Usually we think about that for museums and for artwork, right? So what do I mean by that? I don't know if you noticed on my title slide, there's this logo in the corner, Cell Press, Science That Inspires. Okay, well, logos are logos, but we really like this logo and that tagline because we think it represents well what we do. We're looking for science that inspires the scientific community to keep moving forward. And so really what my job is to try to figure out, what is that? What is the science that most inspires? So let's say you're interested in something like CRISPR. Many people are interested in CRISPR, right? And you do a literature search in PubMed. This is from a little while ago, so that number's a little bit old, but you get something like 26,000 hits. Well, that's a lot of papers. What if I narrow that down? I'm mostly interested in, say, CRISPR papers that are related to gene therapy. Well, if I do that search, I get almost 4,000 papers. That's still a lot of papers to read and sort through. So here, this is doing this search with a tool that we use a lot as editors called Scopus. And so I did the same search looking for CRISPR and gene therapy, but limited it to the last 3 years and only to primary research articles. And doing that, get a little under 2,000 hits. And then in Scopus, I can cite, or I can sort by citations. And so if I do that, we'll see that of these 2,000 papers published in the last 3 years about CRISPR and gene therapy, about 3 of them were cited 200 times or more. Another 7 were cited 100 times or more. 11 were cited 50 times. And then there are about 400 papers that were cited at least 10 times. So 10 citations in a 1 to 3 year period, pretty good, right? But of those, thousands and thousands of papers, about 400 papers are the ones that people were finding useful enough that they were citing it in their own work, right? And of all of those papers, about 10 of them were really highly influential and impactful. Maybe you don't care about CRISPR. Let's take another quick example. Maybe you're more interested in things like the microbiome. If you do a search for microbiome in PubMed, over 100,000 hits. If we narrow that down in Scopus, again, looking at the last 3 years, and only primary research, it's down to a mere 22,000 papers. But if we look at citations there, about 100 papers that were cited 100 times or more, and about 300 papers, 400 papers that have been cited 50 times in the last 3 years. So there's more going on in microbiome research, but still the number of papers that are highly influential is just a fraction of the ones published. And so that's really what we're trying to think about at Cell is, of all the research that's going on in these interesting areas, which ones are the papers that are going to have the most influence on the scientific community, on future questions? What is going to have the most impact? So these examples are based on citations, right, which happens afterwards. And I think sometimes people think all editors care about are citations. And it's really not true. There are other ways that papers can have important impact. So, are the findings of a study of high interest to the general public? Do they help to inform policy decisions? Is the information in the study exceptionally timely? This is something we've seen a lot with research related to COVID-19, right? Things are moving very fast, they're very timely. They help to inform policy decisions that governments are making. They're of very high interest to everyone because COVID is affecting everyone. And so all of these things are relevant for impact as well. And another way that is really important for work to have an influence is it increasing representation and our understanding of less represented groups, of systemic biases in society. That is a tremendously important aspect of a lot of the current ongoing research as well. So I have some examples here on the slide, of news stories about science, and not all of these things were Cell papers. But this is a type of impact that research can have, right? Things that are interesting to the public. And I've also included a tweet there from a scientist talking about a paper from another lab that he finds particularly interesting. And so contributing to that scientific discourse and having scientists talk to each other and talk about what they think is interesting about studies, that's a type of impact. So when we talk about impact, we're not thinking only about citations, but oftentimes citations are a good representation of how the scientific community is taking interest in a particular study or paper. This is just one example of a type of impact that was really important to me. The paper mentioned there in the center of the screen was the first paper that I handled when I started at Cell about a year ago. The first paper that I handled all the way through to publication. And it was a paper that was talking about the epsilon variant of COVID-19, which you may not have heard of because it never really took off. It never caused a major set of cases. But this was the paper that sort of looked at the ability of antibodies to neutralize that virus. And it was featured by Dr. Anthony Fauci at the White House briefing on COVID in the US. So at the time, we were afraid that variant might take off. It was an important paper. It was impactful at the time. I looked last night, it's been cited about 150 times. So it may not end up being the most important study, given that the variant didn't take off, but it was still an influential and important paper. So going back, I mentioned that each journal is focused on its own scope of interest, as well as the conceptual advance in each paper. And so this is sort of a term that editors use a lot, and I'm not sure how much it gets used outside of the editorial sphere. So what is this conceptual advance? What does this mean? To give you sort of a visual representation, I'll use this cover of Cell's special issue from last year. And the artwork here is representing the idea of the present and the future and scientists sort of walking into a bright future, answering questions that are of importance to humanity. And if we just use this sort of as a visual representation and think about what the scientific community knows right now. Let's say we represent that by this scientist that's at the back of the line. And then we think about a new study that has been done. And we say, okay, what did we learn from that study? How much does that move our knowledge forward? Like that much? Or that much? Or a lot? Right? This sort of increase in valuable knowledge coming from a study is the increment of the conceptual advance. So some studies provide a lot of conceptual advance. Some studies provide less of a conceptual advance. But this is one of the things we're taking into account when we decide if we think a study will be influential and important to the field, and to what sort of field it will be most interesting. What is the increment of conceptual advance? So oftentimes conceptual advance kind of gets confused with novelty. And this idea that this is new, this is for the first time, and we'll see cover letters with claims like no one's ever done this before. Well, that can be very important sometimes, but just because something is new doesn't necessarily mean it's advancing the field. And so conceptual advance is really more about how much a study clarifies complexity, answers longstanding questions, clears up confusions in the field, changes thinking about a particular question, or type of problem. And how much it opens up perhaps new directions of inquiry. So beyond just being new, how much it brings clarity and opens up new directions. That's really what we're thinking about when we talk about conceptual advance. So when you send a paper to the journal, the things that we're thinking about are, in general, does it fall within the journal scope? At CEL, the answer is almost always yes, because our scope is very broad. Is it addressing an interesting question, an important question to answer? And then who's it going to be interesting to? To people in the immediate field, to people across multiple fields? And then we get into the data. Only after thinking about those bigger picture questions. And then we'll think about, okay, are the methods here and the types of experiments sufficient to support these claims? And then at the end, based on what we're finding, how much is this advancing the field in terms of concepts? Oftentimes people are concerned that we're paying attention to other things, like what institute the study comes from, if we know the names of the authors on the paper or not, if there are a certain number of authors on the paper. And really, these aren't things that we're taking into account at this stage. We might notice names in institutes, but that's not part of our evaluation process. And whether we know the authors or not, we're going to be looking for the same things in the paper in terms of evaluating its conceptual advance. We're also not focused on things like grammar and formatting at this initial stage. We don't care what format your references are in. If we accept the paper and publish it, that will have to get taken care of later on. But that's not something we're using to decide whether or not a paper goes for review. And same with things like publication fees or things like that. We're really only looking at the science when you first submit the paper. So now that I've talked to you a little bit about conceptual advance, perhaps you're saying, okay, well, how do I make sure my paper has it? Right? And this is generally a question to ask early in the process when you're setting up a study. You want to make sure you're familiar with the literature, that you're asking questions that people haven't already answered, that aren't necessarily in an area where lots of people are currently seeking the answer and might get there before you. But also thinking about things that your field wants to know, questions that other people are interested in as well. Because if other people want to know the answer to the question, they're going to be very supportive of your research. Thinking about what will really make a difference. Not just what's the next question in this series of experiments I'm doing, but what sort of question or study would make a difference to humanity, to society, or to your field. So in general, if you're asking a really small question, you're probably going to get a small answer. And oftentimes a significant conceptual advance comes when you've really designed a study to address big and influential questions. And of course, that relates to resources and all kinds of constraints. But in general, designing interesting studies is the way to find interesting things that will generate a conceptual advance. And so you do that at the beginning of a project and the planning stage. And then as the data comes in, you need to see what that data is telling you and then follow it to really make sure that you continue to address what are the most interesting aspects of the work that you're doing. And then once you've done an amazing study that generates a really interesting conceptual advance, then you have the task of trying to get me to understand it, right? So talking to editors at journals, And so there are a few things to keep in mind when you do this. And so, sometimes you'll be talking to a journal before you decide to submit the paper. We call that a pre-submission inquiry. Or perhaps you're ready to submit the paper and you're writing a cover letter. You want to be clear with the editor of what you think the conceptual advance is. Don't hide that. Don't expect, oh, well, you'll read the paper, you'll figure it out. I will read the paper, but it's really helpful if I know up front what you think this paper is offering. And then you want to highlight that everywhere you can, in the abstract of the paper, sometimes even in the title, in the introduction, in the discussion as you're discussing things. You want the conceptual advance and the way the paper is really bringing something to the field to be a central message that you hit on multiple times in your paper. And as you're talking about this, both in the writing of the paper and in your cover letter, you want to really sort of hone it to reality. You don't want to over-exaggerate what the advance provided by your paper is, but you also don't want to be too humble and underestimate it. Try to really think about what is this study in this paper offering to the community, and then articulate that clearly. So here I'm giving of a cover letter. I think it probably looks terrible on your screen. It's a lot of text. A lot of it is grayed out because, of course, this is confidential. But this is an example of a cover letter that I got last summer that I thought was a really nice example of how you can talk about the conceptual advance of your work. And so you can kind of see here is, they've introduced the paper. They talk about what they've discovered and found, and then mention, there's been an active debate and controversy about the functional importance of these molecules. Here's what we showed. We report a family of these proteins. They're the first enzymes known to specifically produce a thing. So there's some novelty, right? And then mentioning their methods by using crystallography. We identify a molecular mechanism explaining why this particular thing works. And then here summarizing, our study assigns clear biological functions to these particular signaling molecules, ending a decades-old mystery and controversy as to the function of these molecules. So that laid it all right out for me, right? Even without saying the details of what molecules we're talking about, what the overall concept was, you can tell that's a letter that when I read it, I knew what the authors thought was the conceptual advance of their work. So in general, some points about a cover letter. You don't have to know who to address it to. You don't have to know what editor will read it. That's fine. You can just write "Dear Editor." But then write that letter for any scientist to understand. You can assume your editor is a smart and thinking person, but you should also assume that they don't necessarily know all the context of your field. Be clear about what that context is and about the conceptual advance. Be concise, get to the point about things, and that's a cover letter. I don't really encourage you to think about your abstract and what goes into the abstract of the paper. So I mentioned before, editors are all scientists. We've all been working as scientists, so we've all written abstracts. And I know how it goes. Sometimes, you're applying to go to a conference, you have to write an abstract well before your study's done. And so you write a lot about the rationale, maybe something about the methods, not much for results, and then you're done with it, right? And I think sometimes we never really update that abstract. Even when we submit the paper, it's still very focused on the background and not so much about what's in the paper. It's really important to write an abstract that is about that final version of the paper. And the reason it's important is because that's the only thing that some people are going to see. So as the handling editor for your paper, I'm going to read your whole paper. I discuss your paper with my colleagues at the journal. Oftentimes what they'll be looking at is just the abstract. They'll sort of make an impression about the paper based on that, and then we have a discussion. When I invite reviewers to review your paper, that invitation letter has the title of the paper and the abstract. And based on that, they're going to decide whether they say yes and review your paper, or if they say no, they have to invite someone else, maybe someone else, and someone else. And then it takes longer for your paper to be reviewed, right? So having an abstract that really represents what is in your paper and why it's interesting makes a huge difference. So as a quick overview of the editorial process, you probably know this, but just to make sure we're all on the same page, you do some experiments and you write a paper and you send it to editors at a journal. And one possibility is that they'll read it and say, no, thank you, and it gets rejected. And at that point, you have two options usually. You can always talk to a journal when your paper is rejected. You can talk to the editor. If you think that that decision isn't well-founded, you're allowed to talk to us about it. You don't have to just take the no for an answer if you think that there really is something else we should take into account. And oftentimes when we reject a paper, we'll suggest that you transfer it to another journal that we work with. And that's often a pretty good thing to do because we know what those journals are interested in. So when we're referring you to another journal, it's generally because we think your paper would be a good candidate for that journal. So you send in the paper, hopefully it's not rejected. And so it actually goes out for peer review, right? And reviewers then send their comments back to the editor. And at that point, we make a decision. And that decision can be to reject the paper sometimes after review. And in that case, you have those same two options. You might say, I don't really think you should reject my paper. And you continue to have the discussion with us, in which case we might decide to continue to consider it, or we might not. You would have the opportunity to transfer the paper to another journal after that stage and take the reviews with it if it's one of our collaborating journals. Many papers will be invited to revise, and so then it would go back to the authors to address the comments of the reviewers, usually with more experiments, with discussion, and then goes back into the review process. We generally aim to do that only one time, one cycle of revision and re-review, and then make a final decision on the paper. There are some exceptions, but that's usually how it goes. And if the whole process goes well, then we will accept your paper for publication. And at that stage, it sort of leaves my hands as a scientific editor and goes to our production department, production editors. Those are the people who do the copy editing, who check the grammar, who lay out the paper so it looks like a journal formatted article. And that's when we worry about all of those little formatting things at the very end of the process after a paper is accepted. So in terms of the review process, how we choose reviewers is probably the biggest skill of the editorial job, figuring out who's the right person to review your paper. And so we're always thinking about what the technical expertise of a reviewer is, but also things like their broad view of the field. For Cell, we're looking for papers that are going to be particularly influential, particularly interesting to people outside of just a narrow field of inquiry. So we often are looking for reviewers who can kind of speak to that broader perspective as well. And one of the things that perhaps is most surprising to people is that not all of the reviewers for your paper will have the same expertise. So if you use a particular method in your paper, you might have a reviewer who's an expert in that method, but who doesn't do work on the same questions in the same area of biology as you do, because it's covering that angle, right? Whereas we'll also need a specialist on the actual topics of your paper. And so we generally aim to cover all of the angles of expertise on a study, but oftentimes with reviewers who come at the paper from those different perspectives. Typically it's 3 reviewers, but sometimes it's more, sometimes it's less, and it really depends on how many people we need to feel like we have a well-rounded consideration of your paper that allows us to make a responsible decision about the work. And another thing that we're really big on at Cell, that's very important to us, is to have diversity in our reviewer pool. So for every paper, we're usually trying to make sure we have reviewers who are different genders and different geographical areas, at different career stages, just to kind of make sure that we're having a whole diversity of perspectives involved in the evaluation of a study. The job of the reviewers is to really look into the data, assess whether the conclusions that the paper is drawing are well supported by the data, that the experiments are done well and properly, and that they're sort of up to date with the field. And to be able to point out the things that an editor who's not necessarily a specialist in this area wouldn't be able to figure out. And then based on that assessment, reviewers advise us on whether they think the paper is a fit for the journal, on what they think of the conceptual advance and things like that. But ultimately, the decision on every paper It's made by editors at the journal. Generally not one editor. Usually a discussion amongst the editorial team is what leads to a decision. But it's not like a democracy where reviewers vote. And if all the reviewers agree, we publish it. And if they disagree, we don't, or something like that. It really is a process where the editor integrates the perspectives of the different reviewers, who of course have different expertise, and then uses that to arrive at a decision. So if you don't like the decision that was made, the problem is not the reviewers. It's a discussion to have with your editor. And this is a graphic we made that sort of goes into that point that I mentioned. We're really interested in diversity of perspective when we do review. This is an important thing to us at CEL and across CEL Press. In 2019, my colleagues at the journal, I wasn't there yet, made a commitment to increase gender diversity, especially recognizing that the majority of the reviewers were men. And across all of publishing, all of journals, it's a problem that the majority of reviewers have been only men. And so in 2019, we made a commitment to have more women involved in the reviewing process, aimed to have 30%, 33.3% of our reviewers be women. And that's keeping in mind that most papers have 3 reviewers. So the idea was at least 1 reviewer on every paper should be a woman. We did an update in 2021 and looked at the data, and we are indeed doing that. We're a little bit above that 33% right now. And we have papers where all the reviewers are women. We have papers where 2 out of 3, so it's not that we have just 1 woman everywhere. It depends on the field, it depends on the factors that go into evaluating a paper. And of course, there's more than just men and women in the world, and we are interested in involving non-binary scientists in the review process. Some of these things are a little bit harder to track with data, so we don't have numbers for all of it, but we are very much focused on having that diversity in review. And this text I'm highlighting here sort of mentions how our next frontier, which is trying to work on ethnic background and geography in reviewer diversity. And again, these are things that are harder in a way to do, especially when we don't necessarily want to silo people and make decisions based on factors that aren't important for science, but we do want to make sure we have good representation of all the types of scientists in our evaluation processes. All right, so we went through review. You get a decision letter from the journal, and if you ever read a journal's decision letter, you probably know that they're a little strange, right? There's a lot of information in there, and this is an excerpt from something that my colleague Debbie Sweet wrote about, what to do with the decision letter. And she said, yeah, there's this myth out there that the letters are written in code, and we really don't mean for them to be. The letter is telling you the decision on the paper and telling you about possible future steps. And so if you're finding that hard to decipher, it's perfectly fine to ask the editor, what it really means. I would also encourage you to go and have a look at this article. It's on our website called CEL Mentor, which has lots of great resources about the publishing process, things that are very useful oftentimes for people earlier in your career stages, students, postdocs, junior faculty, lots of resources for how publishing works, and also things you might be thinking about in terms of your research. So Google it, CEL Mentor, and you'll see there are all kinds of videos and short articles there with resources. So I mentioned that if you're not pleased with the decision on your paper, you can always talk to an editor about it. So this is what's typically called a rebuttal or an appeal. And it is always fine to appeal. Generally, what we're looking for in an appeal is data-based arguments, things that are really responding to the concerns of the reviewer, particularly with new evidence. Things that are only argumentation are harder. It's harder for us to make a call when there isn't actually something changing about the data in the paper. And so we ask you to, be realistic and only appeal if you really think that the concerns that are raised can be addressed. If what the reviewers are doing is asking you to expand the scope of the paper, to do experiments and things that you don't want to do, it may be reasonable to discuss that more with the journal. It may also be that that paper is just going to be a better fit at another journal. And so that's something that you can kind of keep in mind when you're deciding what to do as your next step with the paper. But the bottom line here is you should never be afraid to write to your editor. And no conversation is final, and like until at the quite advanced stage. So if you really think that there's more to say and more to discuss with a journal about your paper, you should absolutely write to us and discuss with an editor. And at the beginning, I laid out this little graphic, and I said, each journal is thinking about what's within their scope and about conceptual advance. And I'd like to come back to that because each journal is a bit different. They have different scopes, and we also have different expectations for the conceptual advance provided by a study. And so just because your paper is not a good fit for one journal, has been rejected from a journal, doesn't mean it's not going to be an excellent candidate for another journal. And so, when we make those suggestions for transfer, I hope that you'll always see it as a positive thing, and not like, oh, I got rejected, but rather, oh, here's where my paper is going to fit. Because that's how we think about it. We're really anxious to find a good home for your paper and for your research. And so that's why we sort of discuss with our colleagues and try to help the paper find the best place. So these are some graphics, some logos again, but what I hope I've kind of put together for you here is the idea that we're trying to do a high quality process evaluating your work, some curation, trying to figure out what is the most important and influential science, keeping in mind the people involved, thinking about what impact really means in lots of different ways, and ultimately leading to a journal that's full of science that inspires the people reading it, and that inspires future interesting work that keeps propelling humanity forward. So that's what I prepared for you. I think we still have a few minutes for questions, which is great. My contact information is there. I'm on Twitter if you'd like to tweet something, and the journal is also on Twitter. It's the account called Cell Press. There are some other Cell accounts as well, but Cell Press is the journal Cell. So with that, Rosario, I don't know if you're still there. I am happy to answer some questions.
Thank you, Sherry, for the interesting talk. There were many useful advices, and I think also clarify the whole process of reviewing and publishing. I would like to first encourage the audience to write questions in the chat, and then I can read them to Sherry. Yeah, so Sherry, so your video is a little bit blurry, so could you stop sharing the presentation during the Q&A session? Of course, just if you don't want to show something. Okay, but I can start with a question to start heating the motors. So I would start, I guess, with maybe a conflictive question.
Sure.
So it's about negative results. So first of all, if you know or you have a number in mind about the acceptance rate of self-press in general, and if you know the acceptance rate of negative results. And in this line, do you think manuscripts exposing negative results get more frequently rejected, and why? And what are your thoughts in general on publishing negative results?
Yeah, I think that it's a really interesting question. I don't know the statistics off the top of my head about like total acceptance rate. I think at Cell, it's something that we review about 50% of what's submitted to us, which is it's kind of a lot because we get a lot of papers, and about half of those sort of come through review. And I think our overall publication rate at Cell, which again is a very selective journal, is maybe something around 10% of those, but don't quote me on it. I don't have the exact numbers on hand. Other journals at Cell Press would have pretty different ratios about that because each journal is a bit different, but all Cell Press journals are to some degree selective, so that's part of it. To get at the center part of your question about negative data, I think we all know that it's important for people not to keep doing the same experiments over and over that don't work, right? So making sure that negative data is is disseminated, understood, is something that's really important for science. I think the challenge oftentimes for really selective journals is when you just say, well, that's not it, right? Well, we ruled this out. Sometimes it doesn't feel really, satisfying in terms of, well, everyone's going to read that study. And sorry, I have a little siren going on.
Yeah, we are in Berlin. That happens a lot.
Exactly. And so I think it can be challenging when you're talking about the most selective journals and negative data. But there are definitely journals that sell press that are much more open to that. And I would say oftentimes a story that involves negative data ruling something out, but then also ruling something else in. Sort of, it's not this, it's not this, it's not this. Here's all the data about that, but it is this other thing, or this is the direction to go in. Those sorts of papers are always, I think, going to be far more interesting because it puts the record into the literature of like, don't keep doing this, this doesn't work, right? But still opens a new direction, and that can be quite interesting.
Yeah, thank you. So there are some questions in the chat. Stefan is asking, are there plans to do registered reports at Cell?
So I understand the question. Yeah, so this tends to relate to clinical studies, right, and having to register the study protocol in advance. I hope that's what you're referring to. This is something we've talked about. We have collaborating journals at Cell Press, Med and Cell Reports Medicine, they're quite new journals. They were specifically founded, to think about clinical and translational research, and I think they have more specific policies about that. At Cell, we don't currently require pre-deposition, but we are trying to follow the trends in the field, and we're aware of how important that is. So while we don't have a specific policy now, we are trying to be modern in terms of how we think about those questions, and it's certainly something that reviewers are aware of. So if you're doing clinical research and things that are patient trials, I would say it's very important to be up to date with what the expectations of your field are, and the journal is likely to hold you to that as well. In terms of doing our own registration process, I don't think there's any talk about that. We're using the general repositories, things like clinicaltrials.gov in the US, and there are others in Europe as well. We'd be working with those same sorts of organizations for the registration.
Okay, yeah, thank you. Stefan is asking, would it be okay to submit a manuscript that does not follow exactly the journal style, like text formatting, etc.?
Yes, it's fine. So this is, I would say, a really persistent myth that you have to like reformat your paper. We really don't care about the format of your paper in the initial stage. I mean, it does need to be something that kind of looks like the general format of a research article. It needs to have some introductory explanation. It needs to have a good description of your methods. The results should be there and organized and generally into figures and tables. So something that's just completely strange would be a problem. But in terms of making sure that everything is exactly in the right place or follows all the format guidelines of the journal, no, that's not important initially. We're not going to reject a paper based on that. We're really going to be looking at what's the content of the paper, and evaluating based on scope, interest, conceptual advance.
I guess it's just better to do the formatting beforehand, just not to then go again through the entire thing and reformat everything again. That could be a little
Yeah, in general, like if you know that you want to submit your paper to Cell, I would encourage you to look at our instructions for authors when you're writing the paper. You might as well write it in the format of the journal that you expect to go to. But if you've been rejected from a journal and you're transferring to another, you don't necessarily need to spend days changing the formatting. It's okay to submit it the way you initially wrote it. That's fine.
Good. Okay. Noah is asking, are reviewers working full-time or in parallel to the research? How does this interact with the concept of peer review? And is a paper still peer reviewed when reviewers are not doing active research?
Okay, great questions. So I'm not sure that I mentioned that editors at Cell and all Cell Press journals are full-time editors. So we as editors aren't actively doing our own research anymore. This is different from many journals, society journals, where they have an academic editor, someone who does still have a lab and is working as a scientist and also does editorial things. At Cell Press, editors are full-time editors. Our only job is actually, and it's a more than full-time job to do this well. I don't know how academic editors do it, to be honest. But your question said reviewers. So reviewers generally are scientists actively working in that field. We feel that's important because they need to be up to date on all the methods, on the thinking about the data. And so the job of a reviewer does often require them to be an actively working scientist in that field. Do we invite people to review who aren't still actively working? Occasionally, maybe. You know, sometimes an emeritus professor, someone who retired recently, might still be willing to review, and we might work with them. But that's more the exception than the rule. And then we have some people who maybe are working in industry, you know, at pharmaceutical companies, at biotech companies. Again, we don't work as often with those sorts of folks, but oftentimes they have a perspective that's important for evaluating a paper, particularly things involving drugs and pharmaceuticals. And so we sometimes work with them. But for the most part, reviewers are actively working scientists.
Yeah, that makes sense. There is also another question. So is there also something like a quality control of the reviews being submitted? If yes, does this have an effect on whether a reviewer is invited for review again?
So I love that question, and I know why you're asking it, because you know, we all hate the rude reviewers, right? The quality control of the reviews is me, basically, and my colleagues. So there are a few different layers to this. We try really hard to invite people who we know will give a good review. We can't always do that, and especially I talked about, you know, our intents to diversify our reviewer pool, to make sure we're not just asking, you know, the same old people all the time. And so that means we have to invite people we've never worked with before to achieve that goal. And then also, even with people who have reviewed before, you know, scientists are busy. Sometimes they have more time, sometimes they have less. Sometimes they'll feel very rushed, and they'll, you know, write something that's a little bit less thoughtful than other times. So we try really hard to get good reviews for you. You know, get reviewers who will write a thoughtful review. When the comments come in, we generally don't edit them at all, even in terms of typos and misspellings and grammar. It's very rare. You know, each editor has a slightly different viewpoint on this, but most of us really try to just transmit them to you directly, verbatim. And that's partly because we want to make sure that we don't introduce any error into the process. If I correct the spelling of something and I get it wrong, it actually isn't that protein that I thought it was, it's this other one or something, you know, that would be bad. Some of it is because I think it's useful information for you to have. You know, if the reviewer has been more sloppy in their review, that might be useful information to you, and that might be something you want to discuss with me, you know, later in the process. So for the most part, I try to pass on the reviews directly. If someone has said something that seems particularly inappropriate, I usually try to mention that in the decision letter and say, you know, we don't really agree with this particular perspective, or we're sorry that, you know, the phrasing is a bit harsh, and make sure that you understand what our editorial perspective is, and that we don't necessarily share those really harsh comments. But generally, no, we don't soften it for you. We sort of, you know, let reviewers and authors talk to each other as directly as possible in the process. And then, you know, do we not invite a reviewer again if they behaved badly? Yes. But again, it's not like we have a blacklist that goes to all the editors, like, you can never invite this person's bad. We sort of have that for really like severe cases of scientific misconduct. We try to make sure that we maintain that information. But in general, it's a little bit more on the editor to editor level. So if, you know, there's someone who wrote a review for a paper that I was handling, and it was bad, you know, it was just not helpful, it was mean, I don't invite that person again. And I think each editor is kind of like this. We do our best to sort of, yeah, be consistent in that way. But it's one of the biggest challenges of the job. You know, sometimes people who are really doing very top science, who have an important perspective, are not really great at writing a review, and that can be a hard thing to balance, because what they think about the paper is important, but they're not necessarily such a good colleague to the authors who wrote the paper, and that's unfortunate.
Yeah. So, for example, if one reviewer I don't know, they write something that you think is nonsense or something like that. So what does the journal in that case expect us to do? This is my question. It's not written in the chat. Like,
Yeah,
is
no,
it
no,
really
that's a great
necessary?
question.
Really
Thank you
necessary
for
to
following it,
address
because I think
all
this is important.
reviewers, or
I'm sorry, what was that last bit?
if it is really necessary to address all reviewers, like, or we can, for example, ask the editor, like, to talk to the editor to see whether it's really necessary to address that question
So
that maybe doesn't make sense.
sure. Yeah. I think I should be clear that I'm not talking about comments that are bigoted, that are harassment. That's very rare, and of course, that's not really what I'm referring to here. If you think that there's that sort of comment in your review, first of all, I would hope we can kind of block that from ever coming to you. And if it does come to you, then you would certainly want to speak directly to the editor about that particular instance. The majority of these things are just someone being very harsh about not liking a paper, or saying, you've used all the wrong methods here. This is terrible. The worst paper I've ever read. For those sorts of things, as editors, I'm a human person. I have feelings. I'm just like everyone else. But in the job, part of what the job requires of an editor is to be very dispassionate about things. Sort of say, this is what this person says. This is what this person says. Put it together. And that's often the advice I give when you're reading reviews as well. So if you think that that comment is unfounded, bogus, it's wrong, it's based on a misunderstanding, what's most helpful is in your response, explain that as dispassionately as possible. The reviewer is asking us to do X based on what we know, these cited papers. That's not really accurate. That wouldn't, that would be the wrong direction to go. Say that, and sometimes you'll need to have a conversation with the editor in advance, maybe as you're planning your response. If what you're doing is writing a revision and you're responding point by point to the reviews, you can also do that. And we ask you to be very polite to the reviewers, even if they weren't as polite to you. Almost always things go better when the responses are polite and when they sort of acknowledge where the reviewer is coming from, acknowledge that it's a legitimate viewpoint, even if there are some errors involved in it. Giving the reviewer the benefit of the doubt tends to be helpful. But the best way to respond to things like that is by saying what you think about it, and again, like, not your emotions, but the facts of the matter. And that's how we get past it. And that's really the only way an editor can make a decision. It's going to be weighing the different factors and figuring out what makes sense here.
Yeah. Thank you. There is another question from Nils, my supervisor. One feels that lots of papers in the molecular life science get more and more complex, also in cell, with enormous supplements, etc. How do you actually deal with this enormous complexity as an editor, and how can reviewers assess all this information?
Yeah, data bloat, right? It's hard for all of us. And I think there's a stereotype that cell papers are very long, right? We do have one of the longer formats in terms of what we allow. Our research papers are up to 7 main figures, and usually 7 supplemental figures as a maximum as well, which is longer than the formats of some other journals. But not every paper we publish has to have 7 figures. We are perfectly happy to publish shorter papers. Data bloat comes from a few different factors. Some of it is the technology. Now we can do all of this single cell sequencing, omics data sets. It allows us to have a tremendous amount of data. Whether all of that data needs to be in the paper to make your point is a good question. I would say as a journal, we are not encouraging people to always write enormous papers and have enormous data sets. But what reviewers generally want to see is compelling evidence, right? And so as the techniques become more detailed, more advanced, the expectations of the field for what will be shown to back up your claims also increase, right? But that said, we haven't really changed our format. It's still 7 figures, 7 supplemental figures, up to 14 supplemental items when you take into account data tables and things. And we won't let you have more. So you have to find a way to tell a story in those 21 elements, which we think should be enough. But I realize it can be kind of challenging to manage that and figure it out. But please rest assured that we're perfectly thrilled to see a shorter, more succinct, direct paper, as long as it's telling us something that's a really interesting conceptual advance. That's what we're looking for. It's the advance, not how much stuff is in the paper.
Yeah. So there is a last question in the chat. Could you comment on the timeline for manuscripts re-entering through an appeal process? We were told, not at CEL, that such manuscripts have lower priority compared to first-time submissions. Is there any specific reason behind this?
Good question. So I would say that the review process, if a paper comes back after an appeal and we reopen and re-review, generally is similar. There's not usually much of a difference in timeline for that. What is a little bit different is that how long we take to actually work through an appeal. So those are a little bit lower on the priority list. So each day we get an influx of new papers at CEL. It's something between 15 and 30 papers a day, Monday through Friday, every day of the week. And so we have to maintain a pretty good pace of thinking about the new papers and making decisions about them, getting them out to review and making decisions on that. And so appeals can take a lot of time, especially an appeal after review often has a lot of details we need to take into account and think about. And so we don't generally read that the moment it comes in. We sort of put it on a list. We try to make sure it doesn't get forgotten. And it can take us a week, 2 weeks, sometimes 3 weeks. We try not to take longer than 3 weeks, but it may take us that long to get into it. And it's not because we want to make you wait. It's just because there's a lot to do. And so, if a paper is in a field that is highly competitive and there are competing papers out there, and it's really important to move fast, we try to take that into account. And if you tell us that, sometimes we don't know, but if you can tell us what the relevant factors are, we'll do our best to help with that. But yeah, in general, that would be the part of this delay a bit, is then the thinking about whether or not to invite the paper back in. And the perspective is a bit, we made a decision on it. We did what we could. We went through the process. We made a decision. And so what you're doing when you appeal is asking us to reevaluate the decision, which we're perfectly happy to do. But it falls a little lower in priority than making the decisions on newly submitted things. From there, if we say yes, it comes back in, we send it out for review. We handle that in the same way as any paper going for review, as any paper where we invited a revision and it comes back and it goes the same exact process. So there, the timeline depends mostly on how long reviewers take, if reviewers sign on quickly or not. In re-review, if one of the original reviewers declines to look at it again, that can add a little extra time where we find a replacement, things like that. Which, as a note, if you're a reviewer, thank you so much. We appreciate our reviewers. And also, if you're asked to re-review a paper you looked at before, please say yes. Please find time to do it, because it's really hard on the authors if reviewers don't come back and we have to add someone else in at that stage. So I would hope, if you sign on for a paper, please stay with it until the end of the process. It's really helpful.
Okay, there is a last question from Birgit. This is a more personal question. So when in your career did you decide to become an editor, and how did you get the job?
Okay, thanks for that question. Yeah, my career has been perhaps not the standard path to being an editor. Many people sort of decide while they're still in postdoc that they're interested in leaving the bench. And many of my colleagues came into editorial because they loved science, but didn't necessarily love pipetting. And so that's kind of part of what drove them to sort of move into this type of job. I did a postdoc at a pharmaceutical company, which was an opportunity that sort of came to me. I didn't think I wanted a career in pharma, and I was right. And the postdoc helped me figure that out. I was really interested in teaching. I got a PhD because I wanted to teach. I wanted to be a professor. And so I got my first opportunity for a faculty position in South America, in Ecuador. And so I was a professor of medicine, like very young, in a way that in the US I never would have been able to be a professor at that stage, and had a small research program, directed a master's degree program. So I really enjoyed that. But then, that sort of was time limited. And when I came back from Ecuador to the US, I was teaching at a small college that served undergraduate students. And I really enjoyed that as well. But there were expectations for me to do research with very little funding and equipment. It wasn't really a place I couldn't develop a Western blot, but they wanted me to get NIH grants, and so the expectations were very misaligned. There's a lot of competition at small colleges and universities in the U.S. To get that sort of funding, but without the resources. And so I was faced with having to decide what I wanted to sacrifice. If I wanted to sacrifice my standards as a scientist and do less in like research that maybe wasn't so worth doing, but that I could do with some undergrads and publish a paper somewhere here or there once in a while, or if I wanted to really devote myself to teaching or do something else. And while I love teaching, I also was very lucky to have excellent scientific training in a world-class lab, and I felt like I wasn't using my training so much. And so in talking to some friends and some contacts, I had a friend who was working at Cell Press, and I said, would I even be a candidate for an editor job at this point in my career? I'd been a PI and a professor, and she said, oh, I think you'd be great. And so I interviewed for a job at Cell Reports at the time, without even knowing if I wanted it, mostly to get an idea. And when you interview for an editor job, they put you through tests. And so they made me read manuscripts and discuss them and really time limited. The test was they sent me 3 papers and I had an hour, and then I had to call in. It was before we were doing things on Zoom, so no video. I had to telephone call in to the whole editorial team in a meeting and present these 3 papers and discuss them, that I had 1 hour to prepare and talk through them. And then they did another test where they made me read abstracts and make decisions about, do you review it, do you not review it? So it was sort of like this test under fire. And I realized I thought it was pretty fun. Like I liked it. And apparently they thought I was good at it. And so they offered me the job, and I had to decide, am I going to leave? I had a teaching job at the time. Am I going to leave academia and do this or not? It was a hard decision for me to make, I'll be honest. But I took the job, and now I've been at Cell Press for about 3.5 years, and I moved to Cell a little over a year ago. And here I am, going strong. So thank you for that question. Let me talk about myself.
Yeah. Thank you for sharing. Was very inspiring, and yeah, thank you for the complete talk. I think we are now in time to finish the session. And thank you everyone for the questions also.
Yeah, also many thanks from my side. That was really informative, and I really encourage all the young scientists to listen carefully and follow the advice, because I know from my personal experience that this is important. Absolutely. Thanks, Cheryl, for joining.
Thank you all. Thanks very much.
Goodbye.
Goodbye. Thank you.